Hello everyone, this is Dawn Van Dam, and welcome to today's webinar. I'm the Executive Director of 1 Million Solutions in Health, and very thrilled to be bringing to you this presentation for today. We have a very exciting speaker and very interesting and groundbreaking new technology. So let me just give you a little bit of background about what we're doing today, and then we'll whisk right into the presentation. So the title of today's presentation is Disrupting the Radiation Measurement Space, Fracturing Crystals to Improve Accuracy in Drug Discovery. So we're going to go through a little bit of welcome and introductions. We'll do the signature square presentation as we call it, and we'll give you a chance to evaluate what you've been listening to, some closing remarks, and ask you to fill out a survey. And all along the way, please, in the chat box, if you could direct any questions that you have for the presenter. I am underneath the item that says Event Host. So if you just choose me to send me any of your questions, then I will make sure that when we come to the Q&A portion, I ask our presenter, Dr. Stephen Adler, your questions. So our vision at 1 Million Solutions in Health, just to give you a little bit of overview real quick, is to accelerate the discovery, development, and delivery of innovative cures, treatments, and preventative measures for patients around the world. And we do that by shaping healthcare by sharing solutions. We also have, as part of our program, what we call the Hidden Gems program, or an opportunity to get the edge in our marketplace for science, where we help you access the secret vaults of science. And one of those ways is through our partnership with the NIH here. So as you can see here, we reach out to industry. Also, we have this great partnership with the NIH, where we help them look for people to license their technology. We work with academia and entrepreneurs to put those connections together and bring things to market more quickly. So if you are in any of those categories and you'd like us to help you, just let us know. We also have a technology evaluation consortium where we bring together the pharmaceutical and biotech industry and bring them new technologies from vendors or scientists to help them do their drug discovery process more quickly. So again, if you need to access the industry, we can help you do that as well too. And these are just a sampling of some of the people that we've worked with over the years in our technology evaluation consortium, some big names in the pharmaceutical and biotech industry, and just another slide of famous people and important names across the industry and the R&D development area in the pharmaceutical industry. So fundamentally, if we keep it really simple, what we do is find new scientific solutions. We gather input to optimize that technology, and then we share the findings in order to help bring things to market more quickly. So we are a not-for-profit, and that's how we implement our simple process. Why this is important for the NIH, just a couple of items here. As I said, we put every technology through our signature square evaluation process to help understand where its value proposition is to marketplace and also its readiness to get to market. And because we have patents with the technology coming out of the NIH, it's a lot closer to being ready than some other technologies that we're sometimes working on. So what are the goals of the NIH is more licensing agreements, more CRADAs or cooperative research and development agreements. So if something's not yet ready to be fully taken to market, you as the industry can partner with the NIH to help bring it to market and have first right of refusal for those licensing agreements. And then of course they're always looking for more feedback and direction for their future path and input into their roadmap. So that's just a quick overview, and let me just preview our speaker today and the topic. So fundamentally, we're going to be looking at a new dose calibrator for radioactive tracers in biodistribution studies that can vastly increase the accuracy, perhaps even tenfold. So this webinar will introduce a groundbreaking new dose calibrator for radioactive tracers in biodistribution studies that has the potential to advance the technology to a new level of sensitivity, accuracy, and range for small radiation doses. It's a microdose calibrator which is designed to improve radiation dose measurement used in preclinical in vivo and ex vivo radiopharmaceutical drug development studies. It's using a patented concept of a segmented well counter. The radiation measurement upper limits that are usually hindering the use of a standard single crystal well counter have been eliminated in this technology. The current research into this new technology will be presented by Dr. Adler, who I'll introduce in a moment, who has built a fully operational 8-crystal segment microdose calibrator to test his invention. The results of his work will show the potential for the technology to improve accuracy and efficiency of the work done at the preclinical phase of radiopharmaceutical drug discovery. So quite phenomenal. So just a little bit about Dr. Adler. He started his career in medical instrumentation development at the University of Texas Medical School after graduating with a degree in physics from Kenyon College. In 1987, he transitioned into basic physics research when he joined the Physics Instrumentation Master's Program at the State University of New York at Stony Brook. After obtaining his master's, he joined the physics department at Brookhaven National Lab as a software engineer, and then he returned to Stony Brook and joined the physics PhD program from which he obtained his doctoral degree. As a PhD graduate student and then a member of the staff, he participated in a number of high-energy physics experiments and specialized in trigger electronics and data acquisition systems. In 2003, he transitioned back to medical instrumentation when he joined a biotech startup in Boston called Photodetection Systems. His role was to provide his expertise in integrating complex detector systems at the hardware and software levels, and then he led the software development team in developing the software system to operate NeuroPET, the company's entry into dedicated brain PET scanner, and leading the effort in getting the software component of the scanner FDA 510K certified. So a very distinguished career so far, as I mentioned. And then he took his current position with the Lidos Biomedical Lab as the lab director in Dr. Chwaiki's molecular imaging program as part of the clinical monitoring research program. And from there, his focus has been providing support in the PET arm of the clinical protocols and working with the preclinical research teams in ensuring accurate quantitation of the radio tracer experiments, among other areas of research related to preclinical PET instrumentation. His research interests are in the development of radiation detection measurement systems at a small scale to improve accuracy and efficacy of imaging and dose measurements. And I'm going to let him tell you all about the rest. So with no further ado, I'm going to pass it over to Dr. Adler so he can take you through the presentation. And I just realized that maybe I didn't show you a few slides that I was supposed to show you in the beginning. So if I didn't, my apologies for that, but I quickly went through everything, and I'll let Dr. Adler take over the presentation now.
Okay. Well, Don, thank you very much. Can everybody hear me, I hope? Don, can you hear me?
Yes, I can hear you.
Okay, good. Okay, well, you gave my presentation. Thank you.
No, not as well as you can.
That was an amazing introduction. Thank you very much. Okay, so are you going to make me the presenter?
I think you are the presenter already, because did you see any of my slides going by? Right. I think I missed the opportunity to show my slides.
All right, so let's see. I'm going to go ahead and share my screen. And let's go ahead and get this thing up and running. Okay. So, Don, can you see the presentation now?
Yes, perfectly. Thank you.
Okay. Well, I want to thank you, Don, and Joseph Conrad from the National Cancer Institute for helping me get this technology out to to private industry. You guys have really done a great effort in helping me put this presentation together. So I want to thank you ahead of time. All right, so now to start, I'm going to present the work that I've done in building a prototype of a microdose calibrator. And in order to entice you guys to come and listen to the talk, I put a very fancy title to the talk of Disrupting Radiation Measurement Space, Fracturing Crystals, and so on. But it's really, it's a very simple design, but it's very effective. And I'll proceed to show you this. Now, just to give you the 30,000-foot view of my career, Don gave a very good summary, but just to sort of emphasize my background in high-energy physics, I started my career has basically gone up and down the Northeast Corridor. And it started at SUNY Stony Brook in the physics department with a master's in instrumentation program. And it's a very strong physics department, and the reason for it is that it's right next to one of the nation's renowned physics laboratories called Brookhaven National Lab. So I did my work in conjunction with some experiments going on there. Once I joined the PhD program, I joined an experiment called E787, and that's a picture of the detector system and the physics group. And my work as a graduate student was developing the trigger and software for data acquisition of the big detector behind the group of people. Once I got my PhD, I got a staff position, and I moved to another detector system called Phoenix. This was actually centered at one of the intersection regions at the relativistic Cavian collider, which is a proper atom smasher. The goal of this accelerator facility was to recreate the conditions of the universe shortly after the Big Bang by smashing gold atoms together, gold and lead nuclei together. And the detector system behind that group of people was the one used to try to record data from these collisions. And my work there involved, again, doing data acquisition and trigger work for this larger detector system. After 2003, my wife got a job in Washington, so I decided to move on and get into medical physics, and I found a position with photodetection systems up in Boston. That gave me a 6-year period where I had a chance to interface and work in the startup environment, which essentially is a whole other sort of frame of mind in trying to bring product to market. And so anyway, after 2009, the company was reorganized, sold, and renamed, and so on. And I decided that in that whole period of that 6 years, I was commuting from Washington, where my wife had a job, up to Boston and back. I was flying back and forth every week. So I decided enough with that, and the position opened up with Dr. Choiki's program. And I was hired at the National Cancer Institute. Dr. Choiki's program is the molecular imaging program. It's an intramural program with the NCI, and it covers the full range of preclinical to clinical pathways for drug discovery. So it's a small group, but it covers basically the full gambit of working from the ex vivo cell lines through preclinical work with biodistributions through actual clinical trial work that's done. So it really is a great place to sort of submerge yourself into sort of the user end of all this medical technology. So my goal right now is to take everything I've learned in high energy physics, all the instrumentation and technology, and try to bring it to bear on work done in the preclinical space with the drug discovery with radioligands and so on. So that's my 30,000-foot overview of this. And so once I landed in Washington at NIH, working for Dr. Choiki's program, the first thing I did was get into his preclinical lab, where they do all the biodistribution work, and learned about how drugs were actually tested. And the workhorse, or the main method of doing this, is you take a new radioligand, you inject it into a mouse, and you study the distribution of that drug through the various organs and clearances and so on. So these biodistribution studies are a required step for the FDA in order to approve drugs going forward. And we're in our lab, and other labs, they use these radioactive ligands to attach to these new drugs under study, and they go through and measurements of the radioactive agent are done either using a dose calibrator or well counter. And so my initial introduction or exposure to this was looking at these dose and well counters, and I quickly came to realize that one, they're difficult. It's difficult to actually make an accurate dose measurement for these biodistribution studies, especially at the very low doses that are used. Ideally, you want to be able to make dose measurements between 10 nanocuries up to 100 microcuries. But the problem is, this is very prone to error, especially down in the microcurie and submicrocurie ranges, where you have to be working sort of ping-ponging back and forth between the two instruments, the dose calibrator and the well counter. So anyway, so if we go through and take a closer look at these instruments, the dose calibrator is a device that's used to measure fairly high levels of radioactivity, somewhere between 100 microcuries and above, typically used to measure doses in the tens of millicurie range up to hundreds of millicuries. The reason why you can make the measurements of such high activity is that it's an ionizing chamber. So it's basically a chamber full of gas, and then it's held at a high voltage potential, and the radiation, the gamma rays that go through the gas will ionize the gas, and then you measure the current, the subsequent current that comes out. But because it's a gas, it's very low, it has very low efficiency. So to try to measure activity at very low levels becomes very difficult. And there's effectively a cutoff range of about 10 microcuries. Now, a well counter is sort of on the other side of the spectrum. It's another instrument. Instead of being gas, it's actually a crystal, and typically it's a crystal made of sodium iodide coupled to a photomultiplier tube. And it's a single crystal. It's a cylindrical crystal with a hole drilled in it, and you stick your radioactive sample inside. Now, because it's a crystal, it's extremely, has a very, very high efficiency where you can actually count single gamma rays at a time. So you're down to sort of the single gamma ray measurement. Now, the problem with that is you can quickly overwhelm the crystal if you put too much activity in there. So it's good for low ranges, I don't know, tens of nanocuries, hundreds of nanocuries, up to maybe about a microcurie. And then at that point, it's sort of, it stops working because you overwhelm the crystal with too many gammas. And essentially, the crystal starts to glow all the time. So it's a difficult measurement to go higher. Anyway. So, and then I just want to talk about the gamma counters. Gamma counters are essentially well counters, but they're the workhorse for biodistribution studies. And so it's a well counter with a conveyor belt and a robotic arm. And here, what you do is these are containers, and what you do is you fill these containers with the samples, the tissue samples from the biodistribution studies, and you have hundreds of these tissue samples. You put them on the conveyor belt, and then you run it through this machine, and you make all your measurements, all the radioactive measurements of the tissues. Now, one of the issues here is you have to calibrate this instrument in order to get a proper biodistribution measurement. So just keep that in mind as we move forward. So the problems that we have with the current state-of-the-art is accuracy. We have two instruments that we have to work through, the dose calibrator and the well counter. These instruments have an accuracy blind spot, which I'll go through in a minute. Furthermore, when it comes down to the well counter, right, we have issues trying to measure volumes. You have to do, because of the design of the well counter, different volumes of activity will result in different measurements. So you have to do volume corrections, and that becomes very tedious. And I haven't actually seen anybody do those volume corrections in my lab. And finally, the design of the well counter is basically designed to put small volumes in. You cannot measure a syringe. And ideally, you want to be able to fill a syringe with activity and measure it directly. And you can't do that. And I'll talk about that in a bit. Now, to put some numbers to what I'm talking about, I did some initial measurements back when I first showed up, and I saw that I was having these measurement problems. And NIST, the National Institute of Statistics and Technology, is about a 30-minute drive from NIH, from the NIH campus. And I went and I talked to Brian Zimmerman, who's the head of the radiation measurements group there. And I explained to him my issue, and I said, yeah, there's this, what I call this measurement gap, and so on. And he says, well, okay. And he went off and did his own measurements. And so this is data that was collected or given to me by the National Institute of Standards and Technology. So to walk you through this graph, the y-axis is a relative measure of the performance of the device. So if it's 1, means that the device is working properly, making good measurements. Anything outside of 1 means that it's not doing so well. So this is effectively the efficiency of the measurement. And we have data for well counters, for a well counter dose calibrator. And the well counter performs very well at low doses down here. Unfortunately, this is in becquerels, which is the number of disintegrations per second. So to about, at about 1,000 counts to about 10,000 or 100,000 counts, you can sort of make counts per second. You can make a fairly good measurement with a well counter. But it quickly becomes inefficient when you go beyond that. And then with the dose calibrator, you're good with the very high rates, but when you get to the low rate, then again it starts to give you poor data measurements. And this is what I call the measurement gap. And this particular data set, it's about 0.8 microcuries through about 5.4 microcuries, where you've got this blind spot. Now, unfortunately, this is exactly the kind of activity ranges which is critical for doing biodistribution measurements. This is the kind of doses you want to be able to measure accurately. So, so this was a conundrum that I was facing. And so furthermore, the dose, as I said, the dose calibrators, if you look at them, they're actually designed for syringes. If I go back here, the dose calibrator is a long cylinder, and it has this little holder. This holder is actually a syringe holder, or some holes, vials and other things. So it's really designed to do the full syringe measurement. But when you look at the well counters, you can't measure syringes, right? It's not designed to do a syringe measurement. And so if you want to use use a very small dose, you have to then either use dilution methods, or you have to put your dose inside of a small container and then extract it from the container into the syringe. And the more steps you put into trying to prepare your injection dose, the greater the chance of an error. And there's user errors, there's errors in dilutions, there's errors in when you extract solutions into syringes and so on. So it was I just found this to be very problematic. So what I did is I decided to let's sort of wipe the slate clean here. Let's see how we can redesign, or what can we do in order to improve the well counter to make it an instrument where I can actually use it for syringe measurements, volumetric measurements, and have a range which covers properly that microdose measurement level that I'm seeking to make. So so this shows you what I've done. So this is the microdose calibrator. What it can actually do is the design is, the way it works is that it can actually do that larger, it covers that blind spot, and it does increase the accuracy, precision, and reliability, which is what we need for the preclinical studies. And this is a CAD drawing of the first design of the prototype that I made. And I'll go into a little bit more detail about it. The main issue here is the concept of segmentation. So what I've done is I've taken the well counter, I've segmented it into 8 different crystals. The actual IP is defined as any number of crystals. So basically we've taken the well counter, breaking it up into mini crystals, and the reason is a single crystal, when a gamma ray hits the crystal, the whole crystal glows. And that glowing is what actually gives you that measure of activity that's put inside the bore or the sampling well. Now, if you break up the crystal into separate crystals, then you contain that glowing to just a single crystal. And so the rate, the flux that each crystal gets is going to be less, essentially, by the number of crystals that you break the initial crystal up into. So in this case, I have 8 crystals. So each crystal is going to see 8 times less activity. And so the chance for this what we call the pulse pileup effect diminishes, and this effectively extends the range of the measurement system. Okay, so when I did my first design, I figured 8 was a good number to work with. The idea was to see how far I could go with a design of 8 crystals, and I've done a whole series of measurements and tests. And this shows you the assembled prototype that I have and the CAD drawing that we're going with it. And one of the big aspects of this is that it has a nice large bore. The bore diameter is 2 inches. The actual depth is 10 inches, or no, sorry, 10 centimeters. And it can comfortably fit a syringe. And as you can see, one of the issues is, because it's a precise instrument, I've had to develop a couple of these holders, or these support mechanisms, to be able to place the syringe properly inside the bore. But as you can see, I had a lot of fun with my 3D printer, and I have a little holder device there for my syringe. This is a micro centrifuge vial. This is a scintillation glass vial. These are the containers that we use to put the tissue samples for the biodistributions. And all of these fit very nicely inside. So it solved that problem of being able to make direct measurements of the instruments we're going to be using in the biod space. Now, to give you an idea of the block diagram of the instrument that I built, it's, again, like I said, 8 crystals, and I went the main sort of development concept that I went down is I wanted to get as much off-the-shelf technology that I could, so I could just buy the instruments, buy the electronics, put it together, and then write the needed software in order to turn it into a working instrument, to minimize any unknowns, and so technology unknowns. So the most important thing to me was, can I segment the well counter, and how far can I extend the measurement range for the microdose calibrator? So anyway, so I went to Saint-Gobain. Saint-Gobain, with my help, developed this sodium iodide photomultiplier detector module. This is the crystal module that makes up the microdose calibrator. To read out and operate the PMT in these detector modules, I went to a company called Kane. They specialize in high energy physics electronics, which is my backyard. And I got some high voltage controllers and some digitizers to read out the signals. And these are VME based. These are a bunch of VME cards in my VME crate. You can see the cables. This is the high voltage cables and the signal cables that operate the microdose calibrator. And then with some fiber optics, I've got some data feeds into my PC, and I operate everything under some software that I wrote under the PC. So this is essentially how I put this instrument together. Okay. And so now to go through some of the results, so this is the measurement gap, and then if we add the same data from the microdose calibrator, we get this. So what I've been able to do is extend the range of a regular well counter up to about 100 microcuries. Now this upper range is the limit. It's the sweet spot of this microdose calibrator is essentially between this lower end, this 100 nanocuries, down to even as low as 10 nanocuries, up to about 50 microcuries, 60, 70 microcuries. And that is, for my work, is a godsend, because now I've got an instrument which I can measure tissue samples. I mean, for example, a lot of times you do a BioD and the liver has too much activity in it, and it overwhelms your gamma counter, and then you can't measure the liver. Well, I can take my liver sample and stick it in my microdose calibrator, and I can get a very accurate measure of the activity, which is 4, 5, 6 microcuries, and make an accurate measurement for that. And so it just makes my biodistributions so much easier and more accurate. And again, we have this one instrument. I don't have to rely on the dose calibrator to be going and doing dilutions with my dose calibrator. I can just do everything with the microdose calibrator. I make all my measurements. The other set of tests that I did was to see how accurately I could measure different volumes. So I took the extremes in geometry. So I took an insulin syringe, which is the workhorse for our mouse injections, and it's a very thin, it's a 0.5 cc syringe. And then sort of the larger container, which was our glass scintillating vials. This vial down here, this is it holds a volume of about 20 cc's. And then I filled it up with different volumes of activity, and then I plotted sort of the volume versus the activity, and if we're two different isotopes, F18 and technetium-99. And as you can see from the fit, for the F18, I have an R squared of 0.999. For the technetium-99, I have 0.998, and so on. So this extremely linear plots. If you do a further analysis of this, basically this turns down to about a 1% measurement accuracy that you have across a full range of volumes that these two instruments or these two containers have. So the syringe and the scintillation vial. And I have to emphasize there were no volume corrections made. This was the data coming straight out of the microdose calibrator. So there's no volume corrections. The user doesn't have to worry about how many cc's of activity they put in there. And and so that's, again, taken care of with the design of the microdose calibrator. Now, sort of future applications besides BioDs, there are a lot of, the space here I think is opening up. One of the reasons why I dove into this, trying to solve this problem of making accurate measurements at the microcurie level, was I'm working with Dr. Noriko Sato, and she's doing work labeling cells with zirconium oxide. Now, the problem with labeling cells is if you label cells with too much activity, you'll kill the cells. You induce radiation damage, and the cells don't survive. And the radiation limit is about 1 microcurie per million cells, and typically Noriko prepares a couple of million cells for her injections. And I asked her to give me a list of all her injected doses, and I have a histogram of one particular data set of an experiment that she ran, and you can see that she has a lot of these injections in the few to 10 microcurie range. So. And she was using the dose calibrator in order to try to make these measurements, and clearly you're going to start having problems in the injected dose. And you have to have an accurate injected dose measurement in order to make sure that the outcoming data is accurate and correct. So anyway, so she's pushing the limit of the dose calibrator very clearly here. Now, there is a whole field of cell labeling. It's being used for some cancer treatments and cancer therapies and monitoring. So there's a whole sort of new field of sort of opening up that this microdose calibrator can be used outside of just doing regular BioDs. And there's also some work being done with alpha radionuclides, which I'll talk about in a bit. So to summarize, I have a device in my lab which is ideal for doing these preclinical biodistribution studies, and this is a very critical step for developing all new drug agents. And the primary consumer is going to be for the BioD space will be universities and pharma R&D centers. Future therapies are the immune cell, like I was talking, immune cell-based therapies. And then there's also radioisotope-based therapies, which I can go into afterwards if anybody's interested. And Xofigo is one. It's an alpha radioligand. It's radium-223 dichloride that's being used for prostate cancer treatment. And that could prove there's a good possibility that the microdose calibrator can be the instrument that's needed in order to do the dose measurements for the treatments of these alpha-based targeted therapies. So anyway, so that's where there is, I think there is a space for something for use of the microdose calibrator in the clinical clinical space. Okay, so now I want to pass the baton to Joseph Conrad from the Technology Transfer. Joe, are you there?
Steve, I am here. If you can hear me, you won't have to pass me the ball.
Okay.
I'll just have you advance the slide.
Sure.
Okay. Great. So thank you, Steve. So I just want to take the opportunity to thank everyone who's on the call. I'm going to walk you through some of the market analysis that we've done on this product and what the market space looks like for commercial development initially. So we're looking at a preclinical imaging market that in 2016 was about $854 million. And I'm just going to jump to the potential in 2026 for that industry to balloon to $1.34 billion. And as of 2015, if we go to the right side of this slide, as of 2015, these are some of the prominent players in this preclinical imaging space. So some of these names are recognizable, particularly PerkinElmer, Fujifilm Holdings. Let's see who else is down here. Trifoil, et cetera. So these are some of the major players and the countries they actually reside in. But as I point out in the market opportunity section of this slide, in that preclinical space, there are no players that have a single device that measures the doses between the ranges that Steve indicated, which is that 10 nanocurie to 100 microcurie range, with the accuracy and reproducibility of the device that he's developed. So right now, the current standard in the art is to use both the dose counter and the dose calibrator and the well counter to provide those images, and you're still operating in a blind spot area for those specific quantities of radiation that you need to get an accurate measurement in the biodistribution studies. So Steve, if you move the slide to the next one. So right now, we have a pending application that was filed back in 2016, and the commercial advantages of this technology, again, we have a single device that replaces the need for using two separate devices that produces a more reliable and accurate measurement for the biodistribution studies that are required for FDA, moving forward through the FDA. And the dose measurements are designed to particularly cover the gap that is present by using both the dose calibrator and the well counters, which is a major problem for the standard of care, at least in this industry right now. And the other benefit here is that, unlike the prior devices, this can accommodate any sample container, and more importantly, there does not need to be a volume correction associated with the use of this device. So these are some of the benefits of this particular technology that Steve has developed, and I will turn this back over to Steve to close it out. Thanks for your participation. Appreciate it.
Okay, thank you very much, Joe. All right, so future developments. So the proof of principle is done. The the microdose calibrator is it's been designed, put together, operated, and successfully tested in my lab, and we now have it in our preclinical lab. So here is the microdose calibrator as I was installing it next to the dose calibrator here, and you always have to surround these by lead in order to reduce the background. But anyway, so this is sort of the lead cave that the microdose calibrator was being installed into. You can see one of our scanners in the background. So it is operational in the MIP preclinical lab right now. So what needs to be done? Ideally, we need to reduce the production cost. So there's some design refinements. The main issue is you want to replace the sodium iodide photomultiplier detectors with the more current silicon photomultiplier technology. technology. All the rack of electronics, the VME rack of electronics, you want to replace that with some custom FPGA-based readout electronics and replace the desktop with a tablet. That would make a nice sort of standalone desktop or tabletop device that would work well alongside the dose calibrator. And so that's sort of what I see as the future. The other is to segment the crystal instead of 8 into segments either 16 or 32 or a higher number of crystals, which would effectively extend the range from 100 microcuries up to maybe half a millicurie or maybe even up to millicurie. I'm definitely interested in working with anybody who wants to take this further. I can assist with further development, take on my main sort of input to this would be I wrote a lot of software in order to make this thing work, and I would help any commercial partners either take the software or even do further software development or whatever's needed or the testing and to validate the new devices and so on that came out. I'm very interested in seeing this actually make it into other laboratories because, again, I think this is a very useful and necessary tool in order to make all these biodistribution and other radiation dose measurements feasible for others besides myself and at the MIP. So I'm here to help with anybody who wants to take this further. And here's some contact information. Should I throw this back to you, Joe? Joe?
I think Joe has just unmuted himself. Joe, did you want to go through the how to contact?
I just did unmute myself. I was having a little difficulty. All right, so I'm back. So yes, if you have any interest in pursuing this or have questions for Dr. Adler, you can contact Dr. Adler directly. Tom Klaus is the specialist that handles Dr. Adler's technology portfolio, so he's another source of information. He can handle licensing and collaborative component questions, and myself, I can do that as well. But Tom is actually the primary point of contact for Dr. Adler, at least in this particular case. But if you do have questions for me, feel free to reach out to me and I'll be able to answer those questions and or connect you with Dr. Adler, either way. So you've got 3 ways of getting in contact with us. We hope that we hear from you because we'd like to move this technology into commercial development and get it to the researchers who do some incredibly important work for preclinical development of therapeutics. So thanks for your time and attention, and we really appreciate you joining the webinar.
So it's not over yet.
Over the dawn.
Yeah.
No, it's not.
No, we have a Q&A right now. So if you could just send any questions to me, I am Dawn Van Dam, but you would find me by the name Event Host. So if you go into the chat box and just send me any questions, that would be awesome. Also, if you could take a few moments while we're doing Q&A to just go through our survey and answer any questions for the survey, that would also be great as well too. And I'm just going to switch the presenter to myself for a moment, mostly so I can just get you to see Dr. Adler's picture there and see a little bit more about his title and information while I'm waiting for any questions to come in. So thank you very much, Dr. Adler, for a great presentation. And I'm going to start with a few questions that have come in. And I know you have some background slides, so if you need any of those, just let me know and I can pull those up for you. So let me go here to the first question. How do you foresee the development path to commercialization for the microdose calibrator? So could you explain maybe some more of the details about that?
Sure. If you, I do have a slide on, I can help you with that. So I have a backup slide. Do I move the slide forward or how do you?
I can do it for you. Is this it here? Or?
Yeah, I got it. So, hold on.
Can you see my screen? Can you see it?
Yeah, I can see your screen. I'm trying to go back.
You have to tell me, and I'll move it.
So do you see, does a slide say microdose calibrator development path?
No. Let's just see. Right there. Now I've got it.
Yes.
Yeah.
Okay. So the development path, basically, for someone who wants to take on this technology and turn it into a product, would be first to replicate what I've done so far. So take the 8-crystal sodium iodide PMT, the rack of VME, the VME crate, and the VME boards and the PC. Put it together and sell that as a unit. By doing so, you'll be first to market, and you'll be able to generate service support contracts, which is typical for these kind of instruments. And then once you've got that out there and people using it, one of the most important things you do is you get feedback from the customers. And with that feedback, you can start improving the design. And as I said, the first thing would be to increase the dynamic range, change the number of crystals from 8 maybe to 16 or 32, replace the photomultipliers with the silicon photomultipliers, and as you can see, there's the regular PMTs are rather bulky vacuum tube technology, and the silicon photomultipliers are essentially a solid-state version of these PMTs. So you want to use that technology. And then replace all those VME crates of electronics with a single FPGA or a few FPGA or ASICs, essentially miniaturize all that. And then, as I said, take all the software from a PC and put it into a tablet form. And that would be sort of I would think the best path forward to developing the product. So.
Okay, great. Well, thank you very much for that. And if anyone has any follow-on questions to that, just, again, stick them in the chat box to me, Dawn Van Dam at Event Host. I do have a few more questions that have come in. So, Dr. Adler, what do you see as the most important innovation of the microdose calibrator besides extending the dose measurement range?
Well, that's the volumetric, the lack of volumetric, or the lack of a need for volumetric corrections. And if you could go back a few slides, maybe, all right. So do you see the no volume corrections?
Yes.
Okay, so during the development and working on the manuscript for the microdose calibrator, I ran across this one paper that was written back in 1974, where a group of scientists decided to measure what the efficiency of the well counter was as a function of the depth. So what they did is they took a point source and a point source of activity, and they put it at the top, sort of at the bottom of the well, and then raised it a few inches and measured what the efficiency of the well counter was. And what you can see from the arrows is the peak efficiency is a little over 60%, and as you move the point source up to the top, it drops down to a little bit over 30%. That's a factor of 2 in volume and variation of the position of the point source. So this clearly indicates there's a very large volume correction that needs to be made. So if you have a vial that's half full, a quarter full, or completely full, you're going to have to make corrections up to 50% or so, and that can affect in a large way what the result of your biodistribution is. So that's why being able to get rid of that volume correction is so important.
So one of the things that, sorry, go ahead. Were you finished with your?
Yeah. So that's one of the reasons why I built the microdose calibrators I did, with a very large opening, the 2-inch diameter opening, and the 10 centimeter in depth. So it's a very large sampling well.
Great, awesome. Thank you very much. And again, if anyone has any follow-on comments to that, let me know. So one of the things that occurs to me, Dr. Adler, is that either perhaps in your thinking about how this could make a difference, or in your experience, maybe either people you've talked to or in your own lab, have you seen actual examples where this has made a difference, and, or even perhaps can you project that maybe some of the distribution studies have come out in a highly inaccurate way, and maybe that would have been either dangerous or maybe killed the drug later?
Well, here's the problem. No one actually talks about mismeasurement, right? What I did do at one point is there was a fellow that came in from Israel. He was from the Weizmann Institute, and he was working with Enrico doing the radio cell labeling, or labeling cells with radioactivity, with zirconium, the zirconium oxide compound. And I said, and he had to go and measure and calibrate the gamma counter where he was making some measurements. And I told him, okay, so go ahead and make your calibration standard. And the way he makes the calibration standard is by taking a high dose, measuring the dose calibrated, and then diluting it down by a factor of 1,000 in order to get a small sample that doesn't saturate the gamma counter, which is essentially a well counter. So and he did 3 samples. So I took the 3 samples and I put them in the microdose calibrator, and the 3 samples were off by about 50% to 100%. So there were the difference in the actual activity was, I don't know, somewhere between 0.5 and 1.5 and 2. So there was this huge variation in the 3 samples. So one, he has to make 3 samples because these measurements are very inaccurate, and then again, his calibration sample was just was good to maybe 30–40% accuracy. So again, this really shows you how tedious, or how how someone can be open to making mismeasurements. But if you don't have an instrument that can actually show you what the error is, you're completely blind. It's very hard to actually see it. So that was some of the experiences that I had.
Great, awesome, thank you for that. So here's another question that's been sent in. Will your invention affect how regulatory agencies like the Food and Drug Administration and the Nuclear Regulatory Commission, how they oversee radiopharmaceutical drug development?
Well, I did a little bit of research into what the FDA requires, and I came up with these guidances. Do you see the slide that says FDA regulations?
Yep, right here now.
Okay. Now, going through this, there's a whole a whole bunch of paperwork on what you have to do in order to bring a new drug to market. And part of it is you have to do you have to do those BioDs. And in doing those BioDs, what I did is I highlighted this one section here. I don't know how well you can read it. It's number 2. It says, provide an acceptable method of radioassay of the radioactive drug before its use to ensure that the radioactivity calculations actually reflect the administered activity. That's code for you have to make an accurate dose measurement. Now, I think this problem that I've stumbled across is one that people haven't really brought to light, and I think bringing this device to market and once we start announcing the availability in this new technology, it's going to shed light on the fact that we're in an area where we're not making good measurements. And so I think the FDA could come in and see this and start requiring better measurements, or actually prove that you're actually making appropriate measurements in these microdose levels.
Okay, great. Thank you for that. Another question that was submitted is, how will the microdose calibrator integrate into the preclinical radiopharmaceutical drug discovery laboratory?
Okay, well, besides doing those biodistribution measurements, do you have a calibration standard slide? There we go.
Yeah, here's one.
So one of the main issues is you have to calibrate the instruments inside the preclinical lab. And these are the gamma counters that I was showing or talking about earlier. And there's also imaging instruments, PET-CT, SPECT-CT scanners. Now, right now, the way you, as I was saying earlier, the way you calibrate these machines is you use a dose calibrator, you measure a stock, then you have to go through a dilution, and the dilution is a blind step. No matter how well or how confident your the technician or the technologist is, you just don't know if there's a small splash or there's a small error in that. I mean, a small error in doing the dilution can lead to a large measurement error because you're doing these large dilutions of factors of tens, hundreds, and 1,000. So anyway, and then you take that dilute sample and you stick it inside the gamma counter, or you prepare your phantoms and you put into your SPECT scanner, and so, or your PET scanner. So here you can use a microdose calibrator to do that preparation directly without having to do that dilution. And I think that's going to play, that's a big role in eliminating this blind dilution step, and that will improve the accuracy of making these measurements in the gamma counter and the SPECT scanners and these imaging instruments.
Great. Okay, thank you for that. I'm just going to see if there's any more additional questions as we're getting close to the top of the hour. I have one more to ask you, and I'll just see if there's anyone else who has any more after this. So, Dr. Adler, could your invention be used in clinical settings, such as nuclear medicine departments, which are present in most hospitals around the world?
Well, it definitely could. There is one of the new exciting new fields in cancer treatment is the use of alpha radionuclides, and I've got a slide on that I'd like to show. Go over, there we go. So there's a whole new new field in cancer treatment, which involves the use of alpha-emitting radionuclides. And there's a drug called Xofigo that's been, I believe it's produced by Bayer. It's a radium-22 dichloride, and it's used in the therapy in treating bone metastases. Now, what happens is these alpha-based radiotherapies, they're all typical in the fact that you inject very low doses. So for Xofigo, a typical dose is 100 microcuries for a 70-kilogram subject. There's another alpha radionuclide, actinium-225 PSMA, and it's used in the treatment of prostate cancer. Here, the dose is about 200 microcuries. And if you look at the images on the right, this is some of the preliminary data that they've gotten. And you have a patient that is suffering from metastases from prostate cancer. He underwent 2 treatment cycles of this actinium, and you can see how dramatic the effects were in reducing the tumor burden. It's really almost like magic, the way this treatment has worked so well. So there's other therapies that are coming that are based on thorium-227. Again, most of these, and then the thorium, the dose is going to be about 30 microcuries. Now, what happens with these alpha-based radiotherapies is they mostly, the radionuclide produces mostly alphas. It's like 95% alpha emitters, and they produce a few gammas. So if you want to make a measurement of the dose, you only get like 10% of the activity is produced by gammas. So the equivalent, like fluorine-18 dose, would be in the 10 to 50 microcurie range. So now you're starting to get down to the lower limit of what a dose calibrator can measure. And if you're going to be involved in treatment where actually the level of dose that you're providing can you have to make sure you have a very accurate measurement of those treatment doses. The microdose calibrator would become an essential tool, and so this would then be a place where this microdose calibrator would be used in nuclear medicine departments that do these alpha radio-based treatments. So yeah, it could definitely be used in the sort of the nuclear medicine space.
Great. Well, thank you for that information. That was terrific. So again, I'll just put up Dr. Adler's picture here, and just see if there's any last questions. And maybe, Dr. Adler, if you could just think about a few closing remarks. We're just almost at the top of the hour, and I will turn it back over to you and also put up the contact information. So what would you like to say as a summary?
Well, I just want to thank you, Dawn and Joe, again, for you guys helped me bring this technology sort of presented to interested participants in the industry space. My goal is to get this into laboratories so that other people can use this technology. I've had a blast. I mean, I've really, like I was telling Joe, I've scratched an itch that I've had in developing this device. It was a lot of fun. It took me back to my roots as a high-energy physicist, putting this instrument together. But now that I've had my fun, and it's a very useful device for me in my work and in the preclinical lab that I'm in, the future work that I'm doing, I really do think that it's important to get this out to other laboratories, and I can't do that myself. I need an industry partner. The pathway through industry is really the only way to get this technology out to other laboratories that have this need. So, and I'm hoping that someone does sort of pick up the ball at this point. And I'm committed for this. So whoever picks up the ball on this, I'll be right there with you to ensure successful sort of further developments and and distribution into these other labs. So thank you, Don, thank you, Joe. I really appreciate the opportunity.
And well said. Yeah, well said. Because if the scientists can't get it into the medical community and then to get it to help patients, then it's all for naught. So that's why we do this, right? So it's great.
That's it. We want to cure cancer, one step at a time. So that's what we're doing.
Absolutely.
Precisely. So again, thank you everyone for participating today. You have all the contact information here. If for some reason you don't have it or you lose it, you can always reply back on our emails or send me an email, and I'll put you in touch with either Dr. Adler or any of the technology transfer managers. So again, thank you very much. If there was someone who missed this, we will be sending out recordings, and please spread the word because this is great technology and it needs to make it to the marketplace. Thanks everyone, and look forward to the next webinar. Congratulations, Dr. Adler.
Thank you.
All right. Take care.
Take care. Bye now.